morning all. We will be starting renal physiology. I am Dr. Megha Kulshresht. So, starting first with the functional anatomy of the kidneys. Now, after going through this PowerPoint presentation, you will be able to describe the location and the morphology of the kidneys and its gross features. You will be able to discuss the various types of nephrons. You will be able to correlate the histological modifications of the different segments of the nephrons with their specific function. And also, you will be able to differentiate between the cortical and the juxtamedullary nephrons. Now, to begin with the renal system, it is actually known as the excretory system, which constitutes kidney, ureter, urinary bladder, and the urethra. Now, excretory system refers to a system which involves in the removal of the metabolic wastes from our body, like the urea, creatinine, uric acid, and other toxic pigments. Apart from the kidneys, which serve as the excretory organ, the other excretory organs in our body include the lungs, which removes carbon dioxide, the skin, which removes excess of salt and excess of water in form of sweat, the gastrointestinal tract, which removes the undigested waste product. But sensu stricta, the excretory system refers to the renal system. Now, moving to the gross structure of the kidneys, as you all must be knowing that the kidneys are paired retroperitoneal structures which are bean-shaped, present at the level of T12 to L1 vertebra, and they weigh approximately 150 grams and their dimensions are approximately 10 into 5 into 2.5 centimeter. The important part of the gross structure is the presence of this hilum, which is present on this concave side of the kidneys, and it demarcates the site of the entry of the renal artery, exit of the renal vein, and also the site of the renal pelvis. Now, this kidney is surrounded by a renal capsule and also surrounded further by the adipose layer and also the renal fascia. All these things tries to protect the delicate internal structures of the kidney. Now, if we cut the kidney half in a coronal section, the two parts become evident. One is the outer cortex and other one is the inner medulla. Now, the cortex and the medulla contains the nephrons, which are referred to as the functional unit of the kidney. Apart from the nephrons, blood vessels, nerves and lymphatics are also present in the cortical and the medullary portions. Now, medulla, talking about medulla, you can see that medulla is divided into conical masses. You can see this medulla, it is divided into conical masses, and these conical masses are referred to as renal pyramids. Now, the base of these pyramids is lying at the cortico-medullary junction, and the apex of the pyramid is referred to as the papilla. This papilla is opening into small spaces which are referred to as the minor calyx. Now, these minor calyces join together and open into a still further larger unit which is known as a major calyx. 2 to 3 major calyces constitute the renal pelvis. Okay, so minor pelvis, which 2 to 3 minor pelvis join to form the major calyx, and further the major calyx contributes to form the renal pelvis. Alright, now renal pelvis is the point of convergence of 2 to 3 major calyces, and thus the renal pelvis is the funnel-shaped expansion of the upper end of the ureter, and from the renal pelvis the urine flows out into the ureter. Okay. Now, just to revise the structure of the kidney once again, this is the cortex, this is the outer membrane, these are the medulla, which is organized as pyramids. You can see the nephrons which are arranged in the cortex and the medulla. The nephrons are arranged like this. This is an enlarged view of a nephron. and they are arranged in the cortex as well as in the medullary portions of the kidney. The blood supply through the renal artery and the venous drainage through the renal vein is there, and this is the pelvis, which is a funnel shaped extension of the ureter, and the urine finally flows out through the ureter. Now, just to differentiate between the cortex and the medulla, this is an important difference, students, and you might get the difference between renal cortex and the medulla. Cortex contains many glomeruli. However, renal medulla basically constitutes the tubular portions of the nephrons. So, if a nephron is distributed in the kidney, the cortex contain mainly the glomerular portion of the nephron, and the tubular portion of the nephron are present in the medulla. We'll soon be discussing about the parts of the nephron. Main function of the cortex is the filtration of large volumes of blood. Why? Because it is basically consisting of glomeruli. However, medulla is basically concerned with the maintenance of osmotic gradient because it contains the tubular portions of the nephrons. The renal cortical blood flow is large enough, that is 5 ml per gram of kidney tissue per minute, because it needs a constant blood flow so as to filter large volumes of blood. However, renal medulla has relatively low volumes of blood flow so as to maintain the osmotic gradient and to prevent the flushing out of the solutes from the medulla. Think about the functions of kidney. As you all are aware, the main principal function of kidney is the excretion of the waste product. But from the physiological point of view, I would like to make you understand that kidney is not merely an excretory organ, it is actually a regulatory organ which regulates the homeostasis of our body. So the main regulatory organ which regulates the body fluid osmolarity. It is concerned with the regulation of the electrolyte balance, and also it is concerned with the regulation of the acid-base balance. In our further lectures, we will be discussing each aspect of the functions of kidney one by one. So it is a main regulatory organ. Definitely, it performs excretory function and removes some metabolic wastes like urea, uric acid and creatinine, which are obtained by the metabolism of the various products. The kidney also play a very important endocrinal role, which by secreting important hormones like renin. Another important hormone like erythropoietin, you must be well aware of the functions of erythropoietin during RBC genesis. Another important hormone secreted by the kidneys, it is actually not secreted, it converts vitamin D into its active form, that is calcitriol, and this active form of vitamin D helps in the absorption of the calcium. Other endocrinal hormones includes the kinins. So overall we can say kidney maintains the homeostasis of our body. And another feather in the hat is the function of gluconeogenesis. That is, kidney sometimes takes part in the formation of glucose by utilizing the amino acids, but this function is restricted during the processes of starvation. So if there is kidney damage and a patient of chronic renal failure will definitely going to lose all such functions of the kidney, and therefore the patient will suffer from the various disadvantages because of the loss of these kidney functions. So in further lectures we will be discussing these functions in details. Now first, before discussing the physiological functions, let us see the microscopic structure of the kidney which helps it to perform this function. As you all know, nephron, which is known as the structural and the functional unit of the kidney, and there are about 1.2 million nephrons in human kidney. The length of the nephron is about 45 to 65 millimeters, and it is divided into 5 segments, and these 5 segments are histologically different, and this histological difference is dependent on the basis of their function subserved. Alright, and one more thing here to note is that the kidneys cannot regenerate nephrons. Nephrons once damaged cannot be regenerated, but the adaptive ability of kidney is high enough, and therefore with a lesser number of nephrons, kidney is able to perform equal function. So roughly after the age of 40 years, there is loss of 10% of nephron in every 10 years. Now talking about the various segments of the nephrons, the renal corpuscle, which is present in the renal cortex. Renal corpuscle includes the glomerulus and the Bowman's capsule, followed by the proximal tubule, then the loop of Henle, distal tubule, and the collecting duct. Collecting duct is further divided into the cortical collecting duct and the medullary collecting duct. So we can see here the renal corpuscle, which includes the glomerulus and the Bowman's capsule. Then the proximal tubule, which has a convoluted and a straight part, and then the loop of Henle. This is the loop of Henle, leading to the distal tubule, and finally the collecting duct. Okay. Now talking about the renal corpuscle first, also known as the Malpighian corpuscle. It includes two important parts, the glomerulus and the Bowman's capsule. Now what is glomerulus? Glomerulus is nothing but the tuft of capillaries which invaginates the Bowman's capsule. So actually this glomerulus is nothing but the anastomosing capillaries, like a gucha of capillaries which is invaginating the Bowman's capsule. So you can imagine a gucha of capillaries which is kept over a Bowman's capsular layer, like something which is kept over a spoon. So we can compare the Bowman's capsule to a spoon and this tuft of capillaries which is lying over it. Okay. So this tuft has an entry site and an exit site. The entry site from where the blood enters is known as the afferent arteriole, and the site from which the blood exits is known as the efferent arteriole. Right. Now this tuft of capillaries is invaginating this cup-shaped structure which is known as the Bowman's capsule. Okay. Now you can see here this tuft-shaped capillaries. This is the afferent end, this is the efferent end, and this is the Bowman's capsule which forms a cup, and this cup has two layers. One is the layer which is lying just next to the tuft, which is known as the visceral layer or the inner layer, and the outer layer which is known as the parietal layer. So, is it clear to you that the Bowman's capsule has an inner visceral layer which is lying near the tuft, and then outer parietal layer, and this is a potential space between the inner and the outer layer which is known as the Bowman's space. So whatever blood gets filtered, that the filtrate gets collected into this space which is known as the Bowman's space, and then from Bowman's space it moves further into the renal tubule or the further part of the nephron. Okay. Now after the Bowman's capsule, the next segment of the nephron is the proximal tubule. The proximal tubule, you can see this part is the proximal tubular part. It consists of two segments. One is the convoluted portion and another portion is the straight portion. So proximal tubule basically consists of a convoluted portion and a straight portion. It is somewhere around 15 millimeter long. Okay. Straight portion is known as the pars recta. Point to be remembered at this juncture is that the proximal tubule has got tremendous ability of reabsorption. Most of the water, most of the salt gets reabsorbed in this proximal tubule portion, and therefore the histology of this portion, as you can see in this picture, the histologically we can see that this is having a brush border epithelium. Brush border epithelium is present with the microvilli so that the absorptive area is increased tremendously. Okay, moreover, it contains large number of mitochondria towards its basolateral aspect also. Okay, the tubule, the renal tubule contains two sides, has two sides. One is the apical or the luminal side, and other is the basolateral side. So the side which is towards the lumen is known as the apical side, and the opposite side is known as the basolateral side. Proximal tubule has got large number of microvilli, and it is a brush border epithelium on the luminal side, so as to absorb large amount of water and other salts which enters the lumen. So you can see here in this cross section, the in various invaginations are seen on the basolateral side, and the microvilli are present on the luminal side, so as to absorb large amount of substances from the lumen. This is the lumen. Now, after the proximal convoluted tubule, the next segment is the loop of Henle. This is the loop of Henle. Complete structure is the loop of Henle, which is divided into a descending limb, then an ascending limb. Okay, and this loop of Henle, ascending limb, again has two parts: a thin part of the ascending limb and a thick part of the ascending limb. Right, so that means loop of Henle is divided into three parts: the descending thin limb, ascending thin limb, and ascending thick limb. Functionally, the descending thin limb and the thin ascending limb have got similar type of characteristics. They are basically having flattened epithelium, because the absorptive ability is not much, and they not large amount of absorption is not required in this portion. However, the thick ascending limb has got extensive enfoldings on the basolateral side, and large number of mitochondria is present. The epithelium is cuboidal. So point to remember here is that it is descending thin limb and ascending thin limb which contains the flattened epithelium. However, the ascending thick limb contains cuboidal epithelium. Just remember that in most of the places the epithelium is usually cuboidal, but in descending thin limb and thin portions of the ascending limb, epithelium is flattened. So after the loop of Henle, here I would like to say that where this bent is known as the hairpin bent. Okay, so because it's shaped like a hair, so this is the hairpin bent, and from the ascending limb, the next segment of the tubule is the distal convoluted tubule. So after the thick portion of the ascending thin limb, the next portion of the nephron is the distal convoluted tubule. Again, convoluted refers to something which is not straight, and that so from the word it is very much clear that this segment is not a straight segment but a convoluted, a congested kind of a segment. It is about 5 millimeters in length, and the main thing to remember here, it lies in the renal cortex. It is not present in the medulla, it is present in the renal cortex. This portion is less distinct in its brush border epithelium, okay, and the surface area is not too much for any kind of absorption. However, it has the ability for active transfer of substances. Distal tubular cells have poor permeability, which allows them to actively transfer large amount of substances. So that means this segment is characterized by a large number of active channels. Other important part which you need to remember in distal convoluted tubule is a structurally modified segment which is known as macula densa. So macula densa is nothing but a small part of distal convoluted tubule at the site where it just emerges from the ascending limb, and it is a plaque-like thickening, and it is known as macula densa because this macula densa has its own significance in the regulation of blood pressure. We will be discussing it further, the importance of macula densa. Now, from the distal convoluted tubule, arises the next segment, that is the collecting duct. Collecting ducts are formed by joining of the connecting tubules. Now, distal convoluted tubules give rise to the connecting tubules, and the connecting tubules of two or more nephrons join to form the collecting ducts. Correct? Now, these collecting ducts are about 20 millimeters long, and they run both in the cortical as well as in the medullary parts. Right. Now this epithelium of these collecting duct changes from cuboidal in the initial part to the columnar in the later part. The cells have got poor absorptive and metabolic abilities. And finally, in the initial portion, 8 to 10 cortical ducts will coalesce to form a single collecting duct. It will run into the medulla to form the medullary collecting duct, and in the medullary parts, several ducts again fuse together to form a duct which opens into the tip of the renal papilla. So that means collecting duct has two portions, that is the medullary collecting duct and the cortical collecting duct. And two important cell types which you need to remember, which are present in the collecting duct, are the principal cells and the intercalated cells. They take part in the regulation of the potassium balance as well as in acid-base regulation. So in collecting duct, you need to remember that collecting duct is basically having two parts, cortical part and the medullary part. There are two important cells which are present in the collecting duct, that is the principal cell as well as the intercalated cells. So finally, here is a slide which will help you to memorize the histological features of the various segments of the nephron, starting from the renal corpuscle. That this is the glomerulus. This is the Bowman's capsule. You can see here the brush border epithelium of the proximal convoluted tubule. Then this is the thin descending limb of the loop of Henle and the thin ascending limb, both having flattened epithelium. Then here is the thick ascending limb of the loop of Henle, which have got basically the columnar type of epitheliums, and then the intercalated cells and the principal cells, which as I told you, are part of the collecting duct. So finally, just to go through the structure of the entire nephron: renal artery, renal vein, Bowman's capsule, glomerulus, PCT, then the loop of Henle, distal convoluted tubule, collecting duct. I hope the complete structure of nephron is clear to you. If you have any queries, soon we'll be dealing with the queries also. Now, just before we end, I would like to highlight you the differences between the cortical and the juxtamedullary nephrons. A very, very important differentiating question which is asked very frequently in your questions in your exams as well as in your vivas. So nephrons are basically two types, the cortical nephrons and the juxtamedullary nephrons, depending on the location where they are present in the kidneys. Right. The cortical nephrons have got the glomeruli which are located on the outer aspect of the cortex. So just remember that those nephrons whose glomeruli are basically present in the cortex are referred to as the cortical nephrons, whereas those nephrons whose glomeruli are present deep in the renal cortex just near the corticomedullary junction are referred to as juxtamedullary. Juxta refers to near and medullary refers to the medullary area. So in the juxtamedullary nephrons, the glomeruli is present very close to the medulla. So it is not present superficially into the cortex, it is present deep into the cortex just near the corticomedullary junction. 85% of the nephrons are cortical in nature. However, 15% of the nephrons are juxtamedullary in nature. So we have got large population of cortical nephrons and only limited population of juxtamedullary nephron is present. Maximum amount is of cortical nephrons. The glomeruli of cortical nephrons are small, whereas the glomeruli of juxtamedullary nephrons are large. The loop of Henle of the cortical nephrons are short and they penetrate only a small distance in medulla. So you can remember cortical nephrons, small loop of Henle penetrating a short distance into the medulla but having a larger percentage. On the other hand, the loop of Henle of the juxtamedullary nephrons are long and they dip deep into the renal medulla. That means in case glomeruli are small and the loop of Henle, the renal tubular portion is very long and projects deep into the medulla. The vascular supply of the cortical nephrons is through extensive network of peritubular capillaries. So the renal tubule is receiving its vascular supply through a peritubular capillary network, whereas in the juxtamedullary nephrons, the vascular supply is highly extensive through the specialized vasa recta. So you'd have to remember that juxtamedullary nephrons have got a supply of vasa recta. All right. Then the cortical nephrons are have got slow rates of filtration, but the rate of filtration is fast in juxtamedullary nephrons. The major role of cortical nephrons is in the excretion of waste product and filtration, because their loop of Henle are short, but the juxtamedullary nephrons are mainly concerned with the concentration of the urine, because as you see they have got long loops of Henle which are dipping deep into the medulla, and therefore the ability to concentrate urine is larger in juxtamedullary nephrons. More is the length of the tubular segment, more will be the ability to concentrate the urine because of extensive transfer of fluids across the renal tubule. So this is the important slide which presents you the differences between the two types of nephrons. So please, you can see this structure which shows the cortical nephron and the juxtamedullary nephrons. You can see this has got a very long loop, very long renal loop, and here the loop of Henle is short. So I wind up my lecture here, and I hope you are able to understand what I am trying to speak through my PowerPoint presentation. Please go through the presentation, go through the various study materials. You have your books and other study materials, and whatever are your will be your queries, I will be dealing with those queries soon in my further coming lectures. Thank you, students.